So one of the ways we've looked at this is using a lung metastasis model. A lot of cases, primary tumors are reasonably easy to treat. A lot of primary tumors you can actually undergo surgery and remove them. The real risk in a lot of cancers is the metastasis, when the cancer comes back, or the small pieces of the tumor that break off and lodge in other places. So we were modeling this in mice by giving them intravenous injection of cancer cells right into the tail. And then within a couple of weeks, the tumors grow and then the mouse is sick. And then their lungs go from looking like this to looking like this. So this is metastasis of cancer into the lung. So our question was, if we took these lungs and we gave these animals VSV, could we have a positive effect on their tumors? And so we actually used VSV that experiences green fluorescent protein, and then we've actually waited 24 hours and just see if we could see the virus in the tumor. And what we actually saw was this. And you will notice that everywhere there is tumor, there's virus. So that means that the virus has actually got into the tumor, started replicating. And for us, what's really interesting is there's an area in the bottom corner where you see no virus. If you look at the corresponding picture, it's also where there's no tumor. So where there's no tumor, there's no virus. Where there's tumor, there's virus. So that was a very important finding for us. So you can see where the arrow is that there's no virus. And so what we've done is then we looked at how these animals fared if they were either treated with the virus or not treated with the virus. And these are called survival curves. And we didn't treat animals with VSV. They all died within 50 days of treatment. But where we did treat them with VSV, we were able to cure these animals. And so their lungs went from the top to looking pretty much normal again. So this is all well and good. What about something that's actually really difficult to treat? And one of the most difficult cancers to treat are gliomas. And this is a CT scan of a patient with a glioma, and you can see the large white area. That's the glioma. But the dangerous thing about this type of cancer is not what you can see, but what you can't. And so where each of these white dots is where the tumors actually invaded the brain. And this is what makes these cancers highly resistant to treatment, because you oftentimes can't just go in and remove the tumor. So we went back to the mouse, and with some collaborators at the University of Calgary, we were able to actually make tumors that were green. So we looked, so we could follow the tumors and the virus. So these are green human tumors in mouse brain, and you can see on the far left a small green tumor in the brain of this animal. And then we would go in, and we have now a virus that's red, expressing RFP, or red fluorescent protein, and we inject that IV. And then when we look a couple of days later, and this is the microscopy from those brains, we can see both where the tumor is and where the virus is. And what's interesting, or what's important for us to see, is that wherever you see green, you see red, but also at the edges of the tumor, where the tumors tend to invade the surrounding brain tissue, the virus invades with it. So it's not as if it's just getting the main mass, it's also able to spread out into these smaller spaces, which makes it a really great treatment, because it can see a lot more tumor cells than a surgeon can. And so this is a higher magnification of the tumor edge, showing that it does go out. And so that's well and good. You've cured mouse cancer. Everybody is really excited when you cure mouse, but what does that mean to human cancer? And because we're in the Ottawa Hospital, we have access to biopsy samples and surgery samples from patients who come into the cancer center. And so we have the ability to take these tumor samples, create very thin slices, and then put them back into cell culture. So we can then see, well, these viruses actually infect human tumors that have just come out of a human. And what we've seen is that when we take samples, and usually when a surgeon takes a cancer sample, they take a large portion of cancer tissue, but they also take a large proportion of normal tissue. And so what we've been able to see in our tissue slices is that where there's tumor, and we have a pathologist on staff who actually helps us determine what's tumor and what's normal, Where there's tumor, the virus infects really well. Where there's normal tissue, the virus just doesn't seem to be able to do that. And we've been able to show that in many different types of either liver metastasis of breast cancer or a lung tumor mass, and we've shown it in a bunch of different samples. And we, on an ongoing basis, look at human tumor samples. So that's a really great start. And when I came to the lab, my interest was not with VSV and rhabdoviruses. I was actually interested in another group of viruses known as pox viruses. Scientifically, they're in the group Poxviridae. They have a huge genome. It's long, it's double-stranded DNA. When I say large, the typical virus, like VSV, has about 7 genes. The typical pox virus has about 200. So it's so big that you can almost see it in a light microscope. And it also doesn't get into the host nucleus. It actually makes its own nucleus. It gets into the cell, it basically has everything it needs to grow, just churns away, and it destroys the cell. What was interesting to me was that they exhibited what's called a really restricted tropism. So the most famous pox virus ever was smallpox, and we actually eradicated smallpox as a virus. Had killed more people in human history than anything, than any other disease, than any war, than anything. But we were able to eradicate it using vaccination. And one of the reasons we were able to eradicate it is because it only infects humans. It doesn't infect monkeys, it doesn't infect mice, it doesn't infect anything. And other pox viruses are the same. There's a pox virus that infects only mice, doesn't infect anything else. And actually there's a pox virus that only infects camels, doesn't infect any other. There are some pox viruses that have a little bit more of an open tropism. They can infect more than one species. Like, for example, monkeypox can infect monkeys and humans, but by and large, most pox viruses, or many of them, are very restricted. So, and the second reason pox viruses are a really good candidate for oncolytic virus therapy is that vaccinia virus, which is the vaccination strain, has been used in humans at least since 1796. So it's been used in humans for a long time. So we pretty much know what to expect from vaccinia. It's the first virus ever injected into people on purpose, and it has a long, if people think of clinical trials, it has a long history. People were being stopped vaccinated with vaccinia like 1980. So from 1796 to 1980, and they've actually picked it up now, and again in the States. That's a long history. And so vaccinia virus is actually the lead candidate as an oncolytic virus, and I'll show you some clinical pictures. And a lot of the work I've done in my postdoc has been with myxoma virus, and I'm going to show you a couple of really preliminary data, like brand new data with a virus called raccoon pox. So this is vaccinia virus, just so you, this is what he looks like. It's a brick-shaped virion, has a dumbbell shape. This is an electron micrograph. Pretty virus. This is a phase 1 clinical trial using vaccinia virus to treat patients with metastatic melanoma. People usually think of skin cancer as actually pretty benign, but metastatic melanoma is probably one of the most aggressive cancers there are. Once it becomes metastatic, it's a very aggressive cancer, and if this doesn't make you want to put on sunscreen, then nothing will.